for the opportunity to discuss genomic data disparities today. My name is Asmann. I'm Associate Professor of Biomedical Informatics at Mayo College. Last year at ASCO, it was reported that in lung cancer patients, less NGS testings were given to African American or Black patients, as shown in the red rows of this table. That's the clinical practice disparity. My presentation today will focus on another aspect, which is the data disparity. First, I'll describe the findings we published last year in MPJ Precision Oncology about the inflation of tumor mutational burden, or TMB, measured by tumor-only sequencing. TMB high status is now a FDA-approved patient selection biomarker for immune checkpoint inhibitor therapy. However, in the clinic, it is still a common practice to collect only the tumor samples without the patient-paired peripheral blood or germline. Consequently, mutations identified from tumor-only sequencing will need to be filtered bioinformatically using germline variants in public genetic databases such as 1000 Genome, HapMap, Exact, and ESP. The question is, are the TMB values calculated from tumor-only sequencing accurate? TMB is defined as the number of protein-altering somatic mutations per megabase of coding regions by this paper published in 2019. This Nature Genetics paper showed that higher tumor mutational burden predicts better overall survival after immune checkpoint blockade therapy in multiple cancers, highlighted in red boxes. This figure, showing from the left to right, are cancer types, the number of patients included in the study for each cancer, the hazard ratio, forest plot comparing overall survival after ICB between patients with the highest 20th percentile TMB and those with lower TMBs. The bar represents the 95% confidence interval, and the bars to the left indicates better overall survival. The next column is the TMB values from the highest 20% of the patients, show as the cutoff value, as well as the two-sided log rank p-values for the comparison of between high and low TMB cohorts, as shown in the last column. Another study from 2020 has the same conclusion. Patients treated with pembrolizumab had better response rate if patients had TMB high status in blue compared to those with non-TMB high status in salmon color in multiple cancer. We've established the clinical significance of accurate TMB measurements. Now, this paper from Dr. Erin Mansfield's lab at Mayo Clinic from 2020 showed that tumor-only sequencing overestimated TMBs. From the left to the right in the figure are TMBs calculated and filtered using patient-paired germline variants, and TMBs calculated without patient-paired germline sequencing, but filtered using different stringencies of analytic approaches. It clearly demonstrated the overestimation of TMBs in patients with tumor-only sequencing. Based on that study, we hypothesized that due to the underrepresentation of ethnic minorities in public variant databases, TMB will be more inflated in Black or African American patients compared to white individuals with tumor-only sequencing. We used the germline variants and the somatic mutations called in 701 newly diagnosed multiple myeloma patients for this study. 126 out of the 71 patients are self-reported Black. Note that we do not have sufficient data for other minority groups beyond the Black. We applied 4 different approaches to identify true somatic mutations. One, using patient-paired germline variants, what we call GP group. Second, we filtered out all variants reported in 1,000 genome or exact databases. It's called DB0 group. Third, we filtered out variants in 1,000 genome database or exact database with less than 0.1% of population frequency, the DB 0.001 group. Or the last one is the variants filtered out in databases with less than 1% of population frequency, what we call a DB 0.01 group. We observed that in a GP, comparable TMBs between Black and the white group. But if we use database to filter germline variants, we saw inflated TMB in both Black and the white patients, but significantly higher inflation of TMB in Black patients, as shown in this paper, or in the figures. We had similar observations if we look at all genes, the figure on the left, or just the 682 cancer-related genes used as shown in the figure to the right. Note, as shown in the bullet points highlighted in yellow, all variants in the databases were used for the filtering. This is a pairwise visualization of the differences of TMB between race and the variant filtering criteria. This is a 5x5 matrix of pairwise comparison. The red colors are data from Black patients, and the blue colors are data from white patients. The diagonal plots from top left to bottom right, these are the density plots showing the increased separation of TMB values between the race groups while relaxing the non-somatic variant filtering thresholds. And looking at the upper right half of the panels with the numbers, these are the Pearson correlation R values of TMBs across patients. The black numbers are the R values of all patients. If you focus on the numbers in the pink, hot pink box, the TMB from paired germline filtering had much lower correlations with TMBs from public database filtering of any threshold. This suggests that the impact of tumor-only sequencing on TMB estimation varied substantially from patient to patient. We repeated the analysis using only the race-specific variants in public databases, which is to filter the somatic mutations in Black patients using variants from Black individuals in 1,000 genome and exact, and the filtering in white patients use variant from Europeans. The observation is the same. The reason, as highlighted below the figure, is that the minorities are underrepresented in public databases. For example, in 1,000 genome and exact, only 8.6% are of African ancestry, while almost 55% are of European ancestry. We again repeated analysis using a different public database, the ESP 6500. As shown below the figure, the ESP database had about 30% of African American population, a better representation, but the observations is the same. So here's the summary of the part 1. When using public variant database for filtering, non-somatic mutations, TMB inflated significantly higher in Black compared to the white individuals. And second, the TMB as a biomarker for patient selection to receive treatments without patient-paired germline sequencing will likely introduce racial bias. Okay, that's the part 1. Here's the part 2 of this talk, which talked about the racial disparity of sequencing data qualities in TCGA, one of the most widely used databases today. TCGA studied more than 11,000 patients with cancer. 7 cancer patients included at least 50 tumor samples from self-reported Black patients. These 7 cancers are breast, uterine, and the kidney renal papillary cell carcinoma and the renal cell carcinoma, as well as the colon, prostate, and the lung adenocarcinomas. We discovered that 6 out of these 7 cancer types, as shown in the first 6 columns of the figures, had significantly lower number of sequencing reads for both tumor top panel and the germline exome, the middle panel, from Black patients in blue compared to white patients in red. We searched for the source of the sequencing depths disparity. There were no tumor purity differences between the 2 ethnic groups, the bottom panel, based on metadata published by TCGA. Therefore, the higher sequencing depths of white patients did not result from a necessity of deeper sequencing due to lower tumor purities. So Black patients are sequenced with lower depths in TCGA. In addition, the lower sequencing depths in Black patients persisted when we grouped patients, group samples according to sequencing center. TCGA samples were sequenced at 3 different centers. Or when we grouped the samples by specimen collection sites, the observation is the same. There were 18 or 19, I think, tissue contribution sites from the 6 cancer types. This indicated that neither of these factors could explain the depth disparity. Next, we investigated sequencing depths between sample batches grouped by exome capture kits used to process patients' DNA. This is an example in breast cancer. The majority of exomes were processed using 3 versions of Nimbogen exome capture kits. The Nimbogen HG18 exome version 2 kit, the Nimbogen SeqCap Easy exome version 2 kit, and the Nimbogen SeqCap Easy exome version 3 kit. The oldest kit, the HG18 version 2 exome kit, was used to process 65% of white tumor specimens and 23% of the Black tumors, as shown in the top panels, but was used to process 60% of white germline and 23% of Black germline, as shown in the bottom. This significant enrichment of white patients and the significantly higher sequencing depths in this earlier sample batch were likely the reason for the overall higher sequencing coverage of the white exomes in the breast cancer data. Later sample batches using the Nimbogen SeqCap version 2 and 3 kits were enriched with Black patients. Note that sequencing depths did not differ by race processed by the same exome capture kit. Therefore, the imbalanced allocations of Black and white patients among sample batches sequenced at different depths is likely the reason for the overall lower Black sequencing depths in Black patients. This is true for breast, uterine, and renal clear cell carcinomas, as pointed to by the blue arrows. For colon, prostate, and the lung adenocarcinomas, as pointed to by the red arrows, we still don't know the reason. Regardless of the cause of the lower sequencing depths of tumor and germline exomes, it will likely result in less complete data, under detection of both germline variants and somatic mutations, and inferior variant quality among Black patients. An earlier paper, as shown in this left figure, indicated that a minimal depth of 10 to 15 is required for variant and mutation call sensitivity. First, we investigated the consequences of disparate sequencing depths on germline variant calling, focusing on TCGA breast cancer, because breast cancer cohort enrolled the largest number of Black patients and had the biggest racial differences in sequencing depths. We categorized sequencing depths into 3 groups: low, which is below 10 reads, medium is between 10 to 39 reads, and high, which is above 40 reads. Interestingly, the biggest finding in this figure is that even though overall sequencing depths did not differ between Black and white exome within each of the capture kit, as shown in the upper panel, the mosaic plots, the germline exomes from Black patients still had a higher than expected percentage of genomic positions with lower coverage in all 3 kits. This suggested that variant detection could be impaired compared to white patients. Indeed, as shown in the figure B, bottom left, the minor allele frequency, or MAF, of the germline variants located at the low coverage positions was substantially lower than the populational MAFs documented in the public database for both white and Black patients, but more so for Black. In addition, we found that the capture regions were more polymorphic for Blacks compared to whites, as shown in figure C, bottom right. The MAFs of variants located within the exome capture regions was statistically significantly higher among African populations compared to European populations. We speculate that the exome capture probes might be less effective for Blacks because of that. For somatic mutations, it's similar. The tumor exomes from Black patients were enriched with lower coverage positions, as shown in the upper panels. This resulted in the overall lower confidence of somatic mutation cause in Black patients, as shown in the lower panels, which shows subtle but significant differences in 2 quality scores of the somatic mutations. Notably, TCGA made every effort to report all somatic mutations, including those covered by as few as 1, 2, or 3 alternative allele supporting reads. Look at the figure to the left. So, however, even with this strategy of high sensitivity mutation calling, substantial numbers of somatic mutations would still have been missed at the low coverage position. For example, as shown in figure to the right, the lowest panel, assuming 5x coverage, if only 1 alternative allele supporting read is required to call a somatic mutation, only the mutations with B allele concentration, BAC, or the fraction of reads supporting alternative alleles larger than 20% would have been detected. According to BAC overall distribution values recorded by the TCGA mutation reports, more than 50% of the detected somatic mutations had BACs lower than 20%, and therefore would have been missed if the read coverage were less than 5 reads at those positions. This problem was observed in all 6 cancers when we combine the data together. So the summary for part 2. First, we observed lower coverage of both germline and tumor exomes from Black patients in one of the most widely used cancer genomic database, TCGA. Second, the similar observations from samples contributed by different tissue contributing sites or sequencing centers. This is not due to tumor purity differences between Black and white tumors, but this is due to the timing differences of sequencing, for 3 cancer types. So the earlier sequencing batches were concentrated, enriched by white patients, and earlier batches were sequenced deeper. This also has some impact on from the capture kit design. Because of Black patients, Black individuals have higher polymorphisms in targeted region. So we think the capture kit design, which is based on the human reference genome, which is based mostly on white patients, will be less effective for Black patients. The impact of this is the underdetection of both germline and somatic variants in Black individuals. So the conclusion, here's the conclusion for the racial disparity talk. First, the molecular profiling, including the next generation sequencing, is more critical than ever to cancer patients. These tests are not being evenly applied across racial groups, as reported at ASCO last year. In addition, the common clinical practice of not including patient-paired germline sequencing is problematic because germline sequencing can improve the tumor mutational burden determination. And if we use the public database to filter out the germline sequencing, the Black patients are impacted much more significantly due to the underrepresentation of the Black patients in those genetic databases. And we also shown the less quality of sequencing data in TCGA for Black patients. Therefore, the TCGA results needs to be carefully utilized between different racial groups. Thank you.